Hey guys, so today we are going to talk about this thing called PCR, or polymerase chain reactions. This process that scientists like to use to be able to take one section of DNA, or one strand of DNA, and then replicate one part of it, one specific part, many times, so that they can then run a whole battery of tests to understand and to figure out what this single part that I'm showing, the single part of DNA, what's going on with it. We want to know more information about this one part. So basically, we're going to run something called PCR, which happens in a couple of steps. So what we're going to do is run it through PCR, and the first step, which is called denaturing. There's the first step, denaturing, or denaturing. I've heard many different pronunciations. So what happens in this step is we take a test tube and we fill it with a whole slew of stuff. So we take some water, some salts, we take a couple samples of our DNA over here. Then we also put in a ton of primers, a ton of primers, and we throw in some polymerase enzymes as well. And these polymerase enzymes are pretty cool because they have to be able to withstand this extreme heat that we're going to be putting it through. We're going to be heating it up, this test tube with all of our stuff. We're going to be heating it up to about 100 degrees Celsius, about 96 to 100 degrees. So they have to be pretty resilient to withstand that temperature. So once we run it through this first step, what happens is our DNA denatures. So the binds, the nucleotides, break up. So now we have, instead of one strand of DNA, we have two single strands of DNA, which we can then make many copies of. So so the next step we have is called annealing. So what happens in this step is we cool our solution down a little bit to about 50 degrees. I'm going to move this over just so I don't have to draw it again. So we cool it down a little bit, and what happens in this step is our primers from before are gonna bind with a certain section of our DNA, and these are some pretty special primers. To get these, you have to order them specially to fit with a certain sequence of DNA. So in this step, those primers are bonding with our DNA and attaching itself, and then that will lead us into our third step. If I can find the color, find my notes. Let's see. And that'll lead us pretty nicely into the third step of polymerase chain reaction, which is called extension. It's pretty self-explanatory. I'm gonna take our picture and copy it again because no one wants to draw this. It's all nice and planned out. So then what we do is in this step we heat our solution up again to about 72 degrees Celsius. And what happens is, with the help of our polymerase, our primers zoom across the strand of DNA and copy it. So now what we have is our two original strands, single strands of that DNA, and copies, which is pretty awesome, because then what happens? Let me just blow it up, make a bigger picture. So what happens is, now instead of just having one strand, one copy, now we have two copies plus the original strands of DNA. So then what we want to do after this is we're going to run the PCR, polymerase chain reaction. We're going to run it about 25 to 35 more times, just so we can get a ton of samples of this one strand of DNA. And the whole process, after you run it multiple times, takes about 2 hours. So I want to focus a little bit on this exponential growth that's going on, a little bit. But what happens is you start out with your single strand, and here it is, quickly drawn, quickly stretched. And there's your single strands. There's your first copies. Let's connect those. There's your copies in the middle. Then what happens is that single strand then keeps going. That gets copied. Your copy of the top strand gets copied into two more, so now you end up with 4 copies. And so on and so forth. It's just going to keep growing exponentially until you've got a ton of samples with which you can run a whole battery of tests to find out as much information about this section of DNA as possible. This one section over here that I'm pointing to, you can find out as much information about that as you possibly can, which I think is pretty cool. Yeah, so once again, this is called PCR, or polymerase chain reaction. Let me just write that out. Polymerase, that's one word. Chain reaction. And that is an E, not a C. There we go. And there you have it. Polymerase chain reaction, 3 steps. And it's as easy as that.